So imagine this, you are working on a giant puzzle and then spending years trying carefully fitting pieces together. But about when you're just about to finish, things fall apart. Frustrating, right? Well, it's a bit like what happened when scientists try to develop new medicine. So did you know that like up to 90% of new drugs seem working? They actually end up failing when they, are you doing the...
We are studying pharmaceutical science.
Oh, where's about? In
Monash.
Monash, I
Yeah.
used to do that. I did a Bachelor of Monash, Bachelor of Pharmaceutical Science.
Oh, really?
Yeah, Lawrence, right?
Yeah.
She's in my mentor.
Lawrence, she's still there.
Yeah, I'm still there. I still keep contact with her. And then that's me like throwing when like 90% of drugs failing like that. It's mean we're throwing about like $2 to $3 billion, spending like decades to studying for one single drug, only one. Means what like a lot of people is waiting for the cures to the disease. Because you're doing pharmaceutical science, I will ask you think why does that happen? Like why the drug fail when they being developed?
The effectiveness and the toxicity.
Yes, but so that is like the main reason why they fail. But but I think one of the big reasons because they fail on human is because the tool that we use to test them. So like before they give into human, drugs usually like given to in the cells, testing on cells, right? And then they test it on animals. Cells is allowed to see very quick result in like matter of hours or a day. But what is the limitation of working on cells?
It does not reflect the complex body system.
Yeah, because we all know that our body is a very complicated machine. Just think about our body in this cold weather. We have like a goosebump, shaver, and then your brains tell you to put more clothes on, right? So cells kind of tell us all the story, and that's why we need a better tool, for example, testing on animals to give us a bigger picture. The animals is really cool because they similar to us in some way, especially like mice, there's a lot of gene with us, but they're not exactly like us. So the way that they react on drugs may be different to how we react on drugs. So just imagine like building something with Lego. So the pieces may fit, but we're not sure by the end they are telling us exactly the same story. So take example for motor neuron disease, MND. Have you heard about this before? Yeah. What do you know about that? Like not many people actually know about the disease.
So it's a neurodegenerative disease that causes motor neurons to die.
Yeah.
People lose the control over their muscles.
You know, it's a lot. Well done. I need to tell Lawrence that. You may have seen this Binny around or Big Freeze. So they are like fundraising campaign for MND. And MND, just like you said before, they kill the cells in the brain or the spinal cord that actually control our muscle. And think about these cells when they help us to move, talk, or even breathe. The famous patient like Stephen Hawking or Neil Daniher, they actually lose the ability to control their muscle and then they become completely paralyzed within like just few years. And it's very scary disease because there's no cure and at the moment we don't know exactly the cause of it yet. Only like 5 to 10% of it is caused by genetic. So that means any one of us here can have the disease. And in Australia alone, there at the moment more than 2,000 people actually living with MND, and their average life expectancy only like 27 months. The thing that's interesting in MND is that it only affects in humans, not other animals. So that studying MND on animals or testing drug on animals is not really perfect match. And with the MND patient only start showing symptoms when they're like 30, 40, or maybe 50s. So it's actually we cannot make the mouse or animals like mouse living that long. That's gonna be scary if they can live that long. And the other way to study MND is actually look at actual human brains or tissue, but then we can only assess that when they pass away. So that's going to be too late to understand what's going on to stop them, prevent them, or slow the disease down. So we need a better tool to do that. So that is why I'm here. So my name is Chau. I'm second year PhD at the Florey Institute of Neuroscience and Mental Health at UniMelb. I'm on a mission of developing a better tool to study MND. What I'm doing is instead of using one single cell type or animal, I actually put human tissues in a live mouse brain. So this human tissues is actually collected from skin tissue from volunteers, could be any one of us if you're interested, or from MND patients. At the moment, Melbourne Uni have one of the largest human tissues bank in the world, and by transfer like that, we're allowed to, with this human tissues, skin tissue, scientists can transfer them into any cell type in the body. So in this context is the motor neurons, the one that control our muscle. And so with this technique, our human cell can survive in the mouse brain for months, even years, naturally, without any harsh chemical to keep them happy and healthy. And we hope that we will be able to unlock the secrets of the MND or all the human brain disease like Parkinson and Alzheimer's. And maybe next time when you heard about new groundbreaking research, is hopefully, maybe, just maybe, that we're one step closer to solve one of the most puzzling medical mysteries. That's it, thank you. Actually, at the Florey, if you are interested, we are having the open day coming next month. So it literally the only time in the year that we open for public that you can come in, visit our labs and see if any opportunities for students. Like we have some volunteer work. Well, I was from Monash before, and I come to the open day, and that's why I know about that. Otherwise, normally, which year are you at? Third year?
Yeah.
Normally, I think Lawrence will run the intense employability. Yeah. Normally, I will reach out to her. She will reach out to us, and then we will organize a special event for MIPS only.